Yes, ma'am, you can.
Yes, sir. A very good afternoon to one and all present here. I, Dr. Mani Mekle, Assistant Professor from the Department of Biotechnology. I'm profoundly delighted to welcome you all to the 6th day of 1-week professional development program on avenues of biotechnology. This program intends to facilitate the upgradation of the knowledge and skills of the faculty community. The faculty members participate wholeheartedly in this professional development program and share the knowledge with their students to form them aware of the requirements of the industry from work professionals. It is an initiative in the process of learning and fulfilling the emerging needs of universities and colleges. So once again, I welcome you all. I would like to invite my colleague, Dr. G. Priya, an assistant professor from the Department of Biotechnology, to introduce our today's resource person. Ma'am.
Yes, ma'am. Good afternoon to one and all present here for this 6th day of professional development program on avenues of biotechnology. It's an immense pleasure for me to welcome today's chief guest, Dr. Vishnu Kiran, Senior Scientist, IB Group. I thank you, sir, for accepting our invitation for this professional development program. He obtained his graduate in applied microbiology, botany with specialization in nanotechnology from the University of Madras. His field of study and expertise include nanobiotechnology, algal research, aquaculture, vaccine research, bioremediation, and drug discovery services. He has rich experience in research and development and academics for more than a decade. Sir has also practical experience in marketing and corporate communications, human resources, and project management for more than 5 years. He has been certified with Six Sigma and has bagged the Best Scientist Award, Young Scientist Award, elsewhere, Research Excellence Award, and Best Researcher Award for the year of 2021. He has 31 patents to his credit and has also various research publications and book chapters in many reputed national and international journals. He has also served as a book editor in various disciplines such as nanotechnology, chemical sciences, aquacultures, etc. Sir has actively taken part in various research programs conducted nationally as well as internationally. He has been a part of editorial board member in various international journals and also a member of various research forums. On his credit, Sir has successfully steered the responsibilities including resource alignment, technology strategies, and evaluation of innovation management focus areas to achieve the strategic goals during his stint in the senior management role. A skill in enhancing the process operations, optimizing resources and capacity utilization, and augmenting productivity and operational efficiencies while curtailing cost and expenses are highly commendable. High integrity and energetic leader is known for a capability to envision and create SLO, SOP, and various manuals, as well as for recruit and train successful multicultural teams with latest knowledge and modern skills in research and development areas, and also product development fields to develop quality services. Last but not least, an enterprising leader with proven skills in directing personnel towards the accomplishment of a common goal. Once again, I welcome you, sir. Thank you all.
Thank you, ma'am. Now, without further delay, we will turn the time over to our speaker, Dr. G. Vishnu Kiran Manam, to deliver his lecture on real-time PCR and its application. Sir, over to you, sir.
Good afternoon, everybody. Is everybody able to hear me?
Yes, sir. Yes, sir. We can able to hear you.
Are you able to see the screen?
Sir, your screen is visible, but it's blurred. Could you please able to change the mode? It is blurred. Yes.
Now it is okay. Yes.
No, sir. No, sir.
Okay.
Initially, it was fine, sir.
I'll go and do share again.
Okay, sir.
Now it is okay.
We are not. No, sir.
Okay. Can I send you the PowerPoint to Priya so that you can share from your side, madam?
Yes, sir. You can send to any of the co-conveners so that we can share from our end, sir.
Just a minute. I don't know what is the problem because it is here with nothing on my side.
Okay.
What is the problem? It is blurred, madam.
Yes, sir. It's blurred.
Now?
No, sir. It's not clear actually.
Yeah, I've just shared it to Priya. But it is taking time actually.
Yes, sir. We will wait,
Okay,
sir.
meanwhile,
No issue, sir. I request the participant to anticipate for a few minutes. Priya ma'am, kindly coordinate with our resource person, ma'am.
Yeah, sure, ma'am. Sure, ma'am. Wait for a minute, ma'am.
Dear participants, kindly wait for a few more minutes. Our resource person had some technical issues. He will be rejoining soon and he is sending the files to one of our conveners. We will share from our side. Kindly wait for a few minutes.
good afternoon. No.
Sir, would you hear me?
Yes, sir. Yes, sir. Now we can able to see your screen also, sir, clearly, sir. Thank you, sir.
Are you able to hear me, ma'am?
Yes, sir. Yes, sir.
Hello.
It's now fine, sir. It's absolutely fine, sir.
Start. Shall I start?
Yes, sir.
Okay, good afternoon, everyone. Thank you, the SRM Institute for Technology, for giving me this wonderful opportunity to share, the knowledge sharing with everybody. So today, our topic will be.
Sir, excuse me. Sorry to interrupt you.
Basically,
There is some black
yes.
screen which is blocking your screen actually. Yeah, now it's visible, sir. You can proceed. Thank you.
Yes, sir.
Today we are going to talk about the real-time PCR and its applications. So, as everybody there are three types of PCR. So one is the conventional PCR, which was used for almost 3 decades, which means first step we will be having. First, let me tell you what is a PCR. PCR is nothing but a polymerase chain reactions. That PCR is a polymerase chain reaction where a small amount of DNA can be amplified.
Sir,
I-
we are not. So we are not able to hear your voice as well as we are not able to see your screen, sir.
Okay. No, I think I have some problem with my connectivity, madam.
Okay, sir. Okay, sir.
So then no issues, sir. At least you
Just
can just send your presentation to any one of our convenience. We will present from our side. We can just coordinate, sir.
I already shared it to Priya.
Okay, sir. Then wait a minute,
I've
sir. I will
already
ask her
shared
to.
presentation to Priya. No, I will log in with my mobile, sir. Then.
No issues, sir. I will ask Priya ma'am to share, sir. Wait for a minute.
Oh, okay.
Yes, ma'am. We can see you from your screen. We can able to. Yes, ma'am. It's fine now.
Sir, you
Sir.
can proceed, sir.
Are you able to hear me now?
Yes, sir. Yes, sir.
Hello. Phoneல இருந்து லாகின் பண்ணிருக்கேன்.
Sir, can you be able to see my screen, sir?
Yes, ma'am. Yes, ma'am. We can able to see your screen.
மியூட் எடுக்க முடியல. விவோ இருக்கு சார். Hello.
I request the participants to excuse for this technical issues.
Can you hear me?
Yes, sir. Yes, sir. We can able to hear from you, sir.
Can you hear me, ma'am?
Yes, sir. Yes, we can able to hear from you, sir.
Yes, yes. I'm extremely sorry for the inconvenience due to the network issue. We shall continue the session. We'll be having a talk on this real-time PCR and its applications. I think this will be will benefit the biotechnology students in the future also, because the technology has updated itself in terms of real-time PCR. So basically, the PCR is a polymerase chain reaction, which is the amplification of the material in a minimal quantity. So when you have your genetic material or an RNA, so it has amplified so many times the output. For example, give you a forensic science example. For example, if you find at least about,
Sir, excuse
so
me,
in
sir.
that
Sorry,
case,
sir.
the DNA
Sir,
has to be
Vishnu,
converted.
sir. Again, your voice is breaking,
Yes.
sir.
Madam, I
Yes, sir. We can able to hear from you, sir, but the voice is breaking, sir.
Hello. Hello.
Yes, sir. Yes, sir. Yes, sir. My kind apologies to all the participants. We will wait for another couple of minutes. There is a technical issues from our resource person. We will resolve it and we will get back to you. Please.
Hello.
Yes, sir. Yes, sir.
Are you able to hear me, ma'am?
Yes, sir. Now we can able to hear from you, sir.
Okay. So I'll continue the session.
Yes, sir. Yes, sir. You can continue the session, sir. Please, sir.
I kindly apologize the participants due to this network issue from my side. And today we are going to have a session on this real-time PCR. PCR is nothing but a polymerase chain reaction, where a little amount of the genetic material is amplified to a number of times to get the desired output. So the polymerase chain reaction is nothing but the amplification of your genetic material, whether it is going to be your DNA or RNA. Basically, PCR was used, in about 3 to 4, and these only the conventional PCR is used. The conventional PCR is a two-step process where your genetic material is amplified to a certain extent in the first process, and the same genetic material is used. Priya, I'll let you know in changing the slides in between. Don't change the slides. Come to the first slide, please.
Okay, no.
So the first step, DNA is used in the second step to amplify more in the PCR. So now to the advance of technology, real-time PCR is commenced into the place. So the candidates or the students might get confused. What is this RT-PCR? RT stands for real-time PCR or reverse transcriptase PCR. Both these PCR are different. Real-time is different and reverse transcriptase is different. And there's something called as qPCR, which means quantitative PCR. So there are 3 different types of PCR. And now, I think last year we have the technology has launched a digital PCR also. So there are 4 types of PCR now. One is the qPCR, real-time PCR, reverse transcriptase PCR, and the digital PCR. The widely used PCR nowadays is the real-time PCR, where to detect the COVID samples also we use this real-time PCR. Real-time PCR is nothing but whatever is happening in the mission, the amplification part, how many cycles are running, is being displayed into your laptop. Each and every, for example, the PCR cycles are run between 1 to 40 cycles. 1 to 40 cycles is the normal range where the number of cycles is being processed in a PCR machine. So the number of cycles, whatever is happening in the first cycle, second cycle, third cycle, till 40th cycle is being, in real-time basis, you can see it in your laptop or your monitor. Whereas the reverse PCR, in real-time PCR, you can use DNA and RNA as your genetic. In reverse transcriptase PCR is different. The RNA genetic material from the RNA, the complementary DNA is synthesized, and the is RNA based virus. So the is placed into the tube where it is converted into a complementary DNA, and the amplification is first. So these are the main difference between a real-time PCR and a reverse transcriptase PCR. Reverse transcriptase PCR is exclusively used for this RNA as the genetic material, whereas real-time PCR is used for both DNA. It's a matter of whether to use the reverse transcriptase enzyme in case of RNA genetic material. RNA as the genetic material. So next slide, Priya. So before starting a PCR, there are so many prerequisites that we need to follow. For example, we need to do a PCR or the PCR is divided into three types. No, pre-PCR, PCR, and post-PCR. Pre-PCR is nothing but the DNA isolation or the genetic nucleic acid isolation part is called pre-PCR, which is very crucial in your PCR process. Pre-PCR is very crucial part in your PCR process. So pre-PCR, the DNA isolation or the genetic material isolation part should be done properly. The second one is the PCR, where your genetic material is placed into the isolated DNA or the genetic material is placed into the PCR machine to amplify the DNA. Post-PCR is nothing but the gel electrophoresis process, where it is primarily used in the conventional PCR. Now, most of the people, they don't use this conventional PCR, so the gel documentation part is not done in this modern era or modern technology. So in real-time PCR, there will be only two processes. One is the pre-PCR and another one is the PCR. The pre-PCR is the DNA isolation process or the genetic material isolation process. The genetic material could be a DNA or your RNA. In most of the cases, it is in most of the viral cases, it is RNA. There are DNA viruses also, but many of the viruses comes from the RNA background. And the genetic material isolation or the DNA isolation is categorized procedure isolate this DNA material. The more commonly used DNA isolation procedures process. One is the C method, and the other one is the phenol chloroform method. So CTAB and phenol chloroform methods are being used widely for so long, and now it has been replaced by a method, an advanced method called Priya, next slide, please. Hello? Hello?
Yes, sir.
Are you able to hear me?
Yes, sir, but your voice is breaking, sir.
And I can't help it because I'm currently in the terrace and talking to you. So I'll continue.
Yes, sir. Yes, sir. We can continue the session, sir.
Okay. See, the two being used widely are phenol chloroform and the CTAB method. Now these methods are replaced by an advanced method called the silica extraction process. So this silica extraction process, four materials into it. One is the GT buffer, the another one is the DEPC water, the third one is the ethanol, 70% ethanol. So these three ingredients of this silica extraction process. The silica extraction, where the silica, because it has a high affinity to bind the nucleic acids, the genetic materials, leaving behind the proteins, lipids, and other elements apart. So in silica extraction process, the silica is used to, since it has a high affinity to bind the nucleic acids, that is the DNA and the RNA, leaving behind the lipids, proteins, and the other unwanted materials. The DEPC is water. It is nothing but a nucleus-free water, which it will clear the RNase enzyme in the material. So this silica will break the cellular contents and it will help in the DNA extraction process also. So the 70% ethanol, the guanidinium GT buffer is used to break down the cell wall of the any viral particle or any material which is chosen to be as a genetic material. So the GT buffer is used to break the cell wall contents, cell membranes, where the inner contents like DNA, proteins, lipids, everything, is being leaked out of the tube of the cellular membrane. And this silica used in the process binds the DNA and the RNA into the silica material, leaving behind the proteins, lipids, and other materials in the aqueous phase. So this is the basic principle of silica extraction process, which is very useful and an advanced technology. So the real-time PCR is used in diagnostic processes, in forensic applications, and in all other applications. Priya, next slide, please. So in the pre-PCR, the pre-PCR should be conducted in a laminar airflow. It should not be conducted outside. Before conducting a PCR, the PCR room should be fumigated properly, and then it followed by a 2-gram machines ozonation, so that the room is in a notobiotic condition. Notobiotic condition is nothing but free from microorganisms and other unwanted materials. So the room should be fumigated before starting a PCR process, and it should be done using the DNA isolation or the RNA isolation is done using a laminar airflow, and the laminar airflow should be cleaned using ethanol or a reagent called basilol, which is antiviral or anti-parasitic solution. So you clean the surface of the laminar airflow using the ethanol or the basilol, and after that you just switch on the UV light for 10 minutes where the laminar airflow is also in a notobiotic conditions. After that, the DNA isolation process is done. So the pestils also should be used, the pestils for grinding the material or breaking the material should be cleaned with sodium hypochlorite, or it's called as liquid chlorine, and then rinsed with proper tap water and cleaned with and sprayed with ethanol and wiped off with using a sterile tissue paper. These are some of the precautionary measures should be taken, otherwise the DNA genetic material will get contaminated with other factors. So the pre-PCR is a very crucial part in the DNA isolation process to avoid any further contamination. Otherwise, the results will vary accordingly. So in this slide, you see there are two-step process in the real-time PCR or the reverse transcriptase PCR. In real-time PCR, both DNA and RNA can be used as a genetic material, whereas in reverse transcriptase PCR, only the RNA can be used as the genetic material. Slide you can see there is one-step process and there is a two-step process. In one-step process, we use the RNA as the genetic material primers. Primers are nothing but short oligonucleotides, which are short sequences of a nucleic acid material. Short oligonucleotides are called primers. Then you add a reverse transcriptase enzyme, then DNA polymerase, and then the buffer reagents. So the reverse transcriptase it converts the RNA into a complementary DNA part. So the amplification of the complementary DNA starts when the PCR mission runs, and this DNA polymerase will bind the double stranded DNA, or it is called as an annealing process. These everything will take place in a single tube. That is one-step RT-PCR, reverse transcriptase PCR. Whereas in two-step PCR, we have a reverse transcriptase enzyme coming in the first process. For example, the reverse transcriptase enzyme will convert the RNA material into the complementary DNA part. And then in the second step, you add a specific primer related to the complementary DNA. The specific primer is, I already told you, it is a short sequence of oligonucleotides. For example, if you have a sequence of complementary DNA, that is, if RNA using a reverse transcriptase enzyme is converted into a complementary DNA. For example, if the complementary DNA is 8-TGC, so the specific primer is added accordingly so that A will pair with T, G will pair with C. So this will get paired, and the double-stranded DNA will be formed in the process by the DNA polymerase enzyme, which anneals the reactions. So which annealing is nothing but the formation of the double-stranded DNA process. Priya, next slide, please. So this is how the reverse transcriptase enzyme works on one-step process and the two-step process. Everything together in a single tube can be done in a reverse transcriptase polymerase chain reaction. Otherwise, first you add a reverse transcriptase enzyme in the first process where the
RNA is converted into a complementary DNA, and you add a specific primer, which are short sequences of oligonucleotides, and then it will be amplified into a n number of numerous amount of DNA and double stranded DNA by the annealing process, where this annealing process is being done by DNA polymerase enzyme. This is an example of one-step RT-PCR kit, as I have already explained to you. Priya, next slide, please. So this is the primers. I have already told you the primers which is used are oligonucleotides, and this is the flexibility and between the two-step process and the one-step presentation. So what is the difference between the two-step and the one-step? The one-step is more convenient because it is very easy to use, but the two-step gives you a flexible model. Whereas, the one-step, you will be knowing all your RNA genetic material is converted to a complementary DNA into the first step. That is the two-step process. So which gives you a flexible model. So there all the RNA genetic material has been converted to a complementary DNA material. And then in the second process, all the complementary DNA has been amplified using a DNA polymerase enzyme. That is the two-step procedure. Whereas in one-step procedure, everything happens in a single tube. So that is what the one-step process is convenient, but the two-step process is more flexible in knowing the process. Next slide, Priya. Next slide, please. So this is how the amplification part takes place as such. First, the genetic material is broken down. Previous slide. Previous slide. This is the sequence how a PCR. For example, as I told you before, the pre-PCR is more important in terms of DNA isolation, because any error in pre-PCR, that is DNA isolation part, your results will be the false positive results will come. So the DNA isolation should be done in a autobiotic conditions. Your laminar airflow should be free of microorganisms. Your room should be free of microorganisms. All your equipments that you use should be in a sterile condition before taking, before doing a DNA isolation. After doing a DNA isolation, you place your amplified DNA into the PCR machine, where your DNA has been, the single stranded DNA is being, all the single stranded DNA, and you put a primer in the tube. So before amplification, there are some criteria. Priya, next slide, please. Next slide, please. These are the components of PCR. So you have your template DNA. No, previous slide, please. Previous slide, please. Components of DNA. Yeah, components of PCR. So before starting, after the DNA isolation, these are the components of PCR where you will be incorporating into the PCR machine. The first one is the DNA, template DNA. Okay. The second one is the polymerase enzyme, primers, DNTPs, buffer, cations. Not all these are required in a PCR machine. In modern methodology, there will be template DNA is nothing but your DNA isolated. The isolated DNA is your template DNA, and you will have a DNA polymerase enzyme. You will have primers. Primers are nothing but oligonucleotides. You will have a probe. For this modern technology, DNTPs is nothing but, along with DNTPs. DNTPs is nothing but primers, plus you'll have a fluorescent labeled material, which is called as probe. So primer is nothing but oligonucleotides. Oligonucleotides plus a fluorescent labeled material is called a probe. So in modern real-time PCR, these are the four elements that we use. One is the isolated DNA that we are going to process. The other one is the DNA polymerase enzyme. The third one is the primers, which are nothing but short oligonucleotides of the DNA sequence. The fourth one is the probe. Probe is nothing but a DNA fluorescent labeled element along with the oligonucleotides, and you will have another enzyme called, that is tag DNA polymerase. So all these ingredients will be placed into an 1.5 ml Eppendorf tube, and it will be placed into the machine. First, the single standard DNA will be processed by adding the complementary DNA part. So what happens is that this primers what you have added into the machine will be complementary thing to the single stranded DNA. And the DNA polymerase, which is nothing but tag polymerase, this will be helpful in making the double stranded DNA. That is in the annealing process. So the temperature for this annealing and re-annealing process varies depending upon the PCRs, depending upon what is the protocol used. In silica extraction method, the temperatures differ accordingly with the viruses also. So depending upon the viruses, depending upon the genetic material, and depending upon the extraction protocol, the temperatures of the denaturation and renaturation processes varies. For example, if it is going to be a silica extraction processes for a particular virus, the first step, 95 degrees Celsius, will be the first step, and then it will come down to 60 degrees Celsius. These two temperatures are used in all the cycles to get the desired products. Some PCRs use 90 degrees Celsius, then it will come to 62 degrees Celsius, then it will go up to 52 degrees Celsius to get the desired process. So the temperature is designed accordingly based on three criteria: based on the genetic material, based on what is the virus or the microbial nature or whatever the source is being selected, and what is the instrumentation is used, which instrument is used to do the process, PCR process. Next slide, please, Priya. So this is how the process takes place. So we have added, you can see the isolated DNA is there. You're adding primers, tag DNA polymerase, and you're adding the probe, that is fluorescent label. So adding fluorescent labeled element is to know where is the process exactly going on, at which, for example, you can see first the single stranded DNA, the blue color thing is there. Then you can see the primer, the complementary DNA is getting formed, the orange color. So next step, after the denaturation, the extension is going on. See, the orange color and the blue color is getting annealed. So a double strand DNA is formed, and the DNA polymerase, the green color thing, is annealing the process, is sticking the process. The DNA, double strand DNA is formed actually. Next slide, please. Okay, next slide. So as I told you, see, here I've given an example of the temperatures. The first temperature, as you can see, you have two temperatures, 98 and 96. Then you will have the annealing process. Then it goes to 72 and 72. So first, during this temperature, the double stranded DNA is broken down to a single stranded DNA, and the complementary DNA is being the primers is being placed, where this primer will act as the complementary DNA to the single stranded DNA. And this annealing process, after annealing at 72 degrees Celsius, this DNA polymerase helps in binding the double stranded DNA, and the annealing process takes place. Next slide, please. The temperature varies accordingly. Next slide, please. This is everything the same. So how will you confirm your successful PCR reaction? Next slide, please. Next slide, please. See, for confirming a successful PCR reaction, previous slide. Previous slide. For confirming a successful PCR reaction, if it is going to be a conventional PCR, you will confirm it through a gel documentation studies. That is, you have to run a gel, agarose gel electrophoresis, where you'll have a standard, and you'll compare your isolated genetic material band with that of the standard. This is the confirmation for the conventional PCR. In real-time PCR, you will have whatever is happening in the PCR machine will be showed into your laptop. So you will have, there are two things. One is the Vic region and another one is the FAM region. The FAM region will show you whether the isolated DNA material for a particular disease is positive or not. The Vic region will show you whether the DNA isolation, what you have done, is correct or not. So in real-time PCR, there is no chance of error, because whatever the DNA isolation you have done, whether it is correct or not, it is proper or not, it will be showed in a Vic region. The peak will be the peak will show whether the DNA is isolated in a proper manner or not. And in the FAM region, if the peak appears, it will show whether the DNA is positive for a particular virus or not. So the real-time PCR is very sensitive and it is very reliable, so there will be no chance of error. Whereas in conventional PCR, you have to do a gel documentation as shown in the picture here. Next step, please. Next slide. Next slide. Next slide. See, I was telling earlier, what are the precautions to be taken to avoid any error? So, next slide, please. So the DNA isolation part is very crucial. So this is everything the same. See, this slide has been already been told, because the RNA is the genetic material. You're adding a reverse transcriptase enzyme, where the reverse transcriptase enzyme converts the RNA into complementary DNA, and this complementary DNA is being broken down into single stranded DNA. And when you add primers, the primers will act as a complementary DNA to the single stranded DNA, and then this annealing process takes place with the help of tag DNA polymerase enzyme, where this single stranded DNA with the help of complementary DNA by adding the primers will form a double stranded DNA, and this tag DNA polymerase will anneal the reaction, and the desired amplified product is given. Next slide, please. Next slide, please. See, as I was discussing, this is the peak, real-time PCR peak. There will be peak like this. For example, when you see there is a Vic region and a FAM region, I told you. When you get a peak like this in the Vic region, it means your DNA isolation is very good. You have made no error in your DNA isolation part. For example, the peak should come like this. For example, if you're isolating 5 types of DNA, so the peak should come like this. This is a good example of a good DNA isolation. For example, in a FAM region, if you get a peak like this, it means it is positive. For example, I'll tell you, next slide, please. Next slide, please. All these been discussed. Next slide. Next slide. So this DNA binding dice is nothing but the FAM region and the Vic region. Tagman probes, the FAM and these are nothing but the dice which have been added to the PCR template. Next slide, please. There are different types of dice which are available for this. There is something called a cyber green. There is FAM. There is Vic. For Vic is something in yellow color. FAM is something in green color. Now all these things are added. For example, you have something called as cyclic CT value. CT value gives whether your DNA sample is positive for a virus or not. For example, when we run 1 to 40 cycles, if you get a peak before 35, before 35th cycle, that means your sample is positive. And if you get a peak after 35, that means your sample is negative. For example, when you're testing for a COVID-19 sample, okay, and you take a nasal swab and you isolate a DNA from that, and when you run the PCR machine, and if you get a CT value of below 35, for example, if you get a CT value if it is going to be CT value is nothing but cyclic threshold value. If the CT value is 17, it means it is positive. If the CT value is 25, it means it is positive. If the CT value is 33, it means it is positive. If the CT value is 37, it means it is negative. If the CT value is 16, you have more infectivity. The infection rate is higher in terms of COVID. For example, the lesser the CT value, the higher the rate of infection. For example, if you get a CT value of 10, you have virus. It is below 35, you're positive, but your level of infection is very less. So the lesser the CT value, the higher the infection. And if your CT value is above 35, it is negative for the virus particles. This is how the PCR mission is validated. Next slide, please. So these are some of the controls in real-time PCR, non-template control. So when we do a real-time PCR mission, we do have to put, unless and until you're very familiar, you're very experienced with the real-time PCR mission, maybe 5 years you're working with a PCR mission or 10 years you're working with a mission. Unless and until you have more practical experience, you need to have some controls while running a PCR mission. For example, we'll be making a positive, we'll be having a positive control of the same virus. For example, if you're going to do a COVID sample, we'll be having a positive control, positive COVID samples, and we will be diluting this positive control from 10 to the power of -1 to 10 to the power of -5 dilutions. So we'll have a two positive controls when we are running a unknown DNA. For example, I'm taking a nasal swab from somebody who's suspected to have a COVID. So that is your unknown sample. And I'll be having a two positive samples of COVID samples. I'll be adding 10 to the power of -1 and 10 to the power of -5 in one of the columns in PCR mission. So, and third column, I'll be having this unknown sample, isolated DNA. And fourth column, I'll be putting a negative control. A negative control is nothing but you can add a DEPC water, or you just can add a master mix. Master mix is nothing but a combination of premix and a template DNA is called as master mix. So you will have two positive controls, that is 10 to the power of -1 positive control and 10 to the power of -5 positive control. In the third column, you'll have unknown DNA isolation, and fourth column, you'll have a negative control. So when the PCR is completed, you can compare the unknown the DNA isolation with that of the positive control, and you can always see how much is the level of infectivity comparing with the positive control also. This is one methodology of comparing the level of infectivity with the positive control. Otherwise, it is very easy in real-time PCR depending upon the CT value obtained. The CT value, if it is lesser, the lesser the CT value, the higher the level of infection. Next slide, please. Next slide, please. So the applications of real-time PCR, the real-time PCR can be used in various applications. It can be used in aquaculture, it can be used in medical diagnostics, it can be used in forensic applications, it can be used, in all the things, in building up a new diagnostic kits. Whatever it is, real-time PCR is very useful because it is very specific and reliable in terms of the results, not like the conventional PCR, where you can do errors in the gel documentation parts comparing with the standards. Here in real-time PCR, whatever is happening in the mission in each and every cycle is being displayed into your computer monitor or your laptop. So what is happening in the first cycle, what is the level of peak you're getting in the first cycle, what is the level of peak you're getting in the 15th cycle, everything is being displayed into your laptop. So you can always screen for viruses in aquaculture like WSSP, white spot spirit syndrome, enterocytozoan syndrome. In molecular diagnostics, you can always screen for this hepatitis B, hepatitis viruses, RNA viruses, COVID viruses. It can be used as a very good diagnostic tool. So these are the very essential part of this application of real-time PCR in terms, seen come across the pandemic, and this real-time PCR mission was the ultimate tool which was useful to the human mankind in screening this COVID samples and in saving the lives of people, whether telling them what is the level of infectivity also. Next slide, please. Next slide, please. So that is all about the real-time PCR. I will just conclude the session again by telling you some essential points. Don't get confused between a RT-PCR is a similar terminology for real-time PCR as well as reverse transcriptase PCR. In real-time PCR, you can use DNA and RNA as a genetic material, whereas in reverse transcriptase PCR, RNA is used as a genetic material. In reverse transcriptase PCR, RNA genetic material is converted to complementary DNA with the help of reverse transcriptase enzyme, and this complementary DNA is amplified many PCR machine where the single stranded DNA is made into a double stranded DNA using the primers and the DNA polymerase enzyme, which is nothing but the tag DNA polymerase enzyme. Whereas in the real-time PCR, both the DNA and the RNA material can be used as the genetic material. The most important criteria in real-time PCR is your DNA isolation process. The three types of procedures, extraction protocols can be used. One is the CTAB method, one is the phenol chloroform method, and another one is the silica extraction method. The silica extraction method is very beneficial, replacing the older methods of CTAB and phenol chloroform methods, because the silica has high affinity in binding the DNA and the RNA, leaving behind the lipids, proteins, and other unwanted materials in the nucleic acid isolation processes. The GT buffer used in the silica extraction processes helps in the breakage of cell membrane or the cellular contents, whereas the same function is being done by the SDS in the phenol chloroform extractions. So SDS is nothing but a similar kind of a detergent, whereas this vanadium thymidine GT buffer is very strong agent in cleaving the cell wall and the cell membrane of the viral particles. So silica extraction process is an advanced technology in the DNA isolation or the genetic material isolation process, and the various applications of the CT value is a main criteria in determining the level of infectivity also. The lesser the CT value, the higher the level of infection. The more the CT value, the lesser the level of infection. And as again I told you, there are various dyes which we use in the real-time PCR like cyber green, FAM region, WIC region. WIC region is we use it for to identify whether the DNA isolation what you have done is proper or not. And the FAM region, if the peak comes, it shows you whether it is positive or not. If the peak doesn't appear in the FAM region, it means it is negative for your genetic material, whatever the diagnosis you're doing. If you don't get any peak in the WIC region, that is VIC region, your DNA isolation is wrong. You should get a very beautiful, as shown in the figure previously. That means you have a proper DNA isolation. If you don't get any peak, and if you get a disorganized peaks in your WIC region, that means your DNA isolation is wrong, and you have to repeat the experiment. And if a peak doesn't appear in the FAM region, it means your sample is negative, and a peak appears in the FAM region below 35 cycles, it means it is positive for the region. So I will conclude the session. I thank Priya and other conveners and the SRM Institute of Technology for giving me this great opportunity to share my knowledge on the real-time PCR. I thank each and every one of the participants who were, listening patiently and who bared with me for my, inconvenient this network issue. I'm extremely sorry for, for this network issue. Thank you so much. Have a wonderful day ahead.
Yeah. Thank you, sir. Thank you so much for this insightful presentation. And now the session is open for the discussion. Participants can put your question in the chat box. I request the participants, if you want to discuss with our dignitary, kindly put your questions in the chat box.
Ma'am, actually, one candidate have asked whether there any training has been giving in this industry for a female candidate who gets a hands-on experience on RT-PCR. So you can
for
ask with
freshers,
sir whether it's
So for freshers,
Sir,
any opening is there, sir?
I'll tell you, I'll answer this, sir. The training is mostly will not be given, sir, because the instrumentation of real-time PCR is around 20 lakhs. And one of the most important factors for this avoidance of this training is that the biosecurity issue. So most of the people coming from outside will carry some other types of, microorganisms or carriers. The training can be given using a proper biosecurity measures. For example, taking a bath before entering the PCR room, and washing your hands with this sanitizers, and you should be completely sterilized before entering into the PCR machine when you're going to do the utmost procedures. One is the exact too much cost of the machinery. People are afraid, what will happen to the machinery if it is given to the learning procedures. That is one difficulties is being placed. But this real-time PCR, you could, they are ready to show what is the process. One thing has been done by RGCA, Rajiv Gandhi Center for Aquaculture at Sirgali. They will be giving real-time PCR demo yearly, once a year. So whenever I get that information, I can pass it on to the group for this real-time PCR training. Any other questions, sir?
Yes, sir. How CT values are fixed? There is a question from Dr. S. Subaranjani.
Sir, CT values are not fixed as those, sir. See, CT value is nothing but a cyclic threshold value, where when you get a peak, for example, when you run a real-time PCR machine, we run for 40 cycles. To 40 cycles. So each cycle, this process takes place. Breaking of the genetic material into a single stranded DNA, then the polymerase chain reaction will occur. The primers will come into takes place, and the complementary DNA will form. And the annealing process will take place. The DNA polymerase will anneal the reaction into a double stranded DNA. Each and every cycle, this process takes place. So when you, for example, the CT value is nothing but the cyclic threshold value. When you get a peak at the 16th cycle, when you get a beautiful peak as shown in the previous figure, in the 16th cycle, that means your sample is positive for a particular disease. And if you get a peak cyclic threshold, CT value in a peak at after 35th cycle, it means it is negative. That is what I told you earlier. When you get a peak, very fast, between 1 to 10 cycles, your level of infectivity is very high. The infection of the disease is very high. Between 10 to 20, it is somewhat okay. 20 to 35, your level of infectivity is low. So whenever you get a CT value, at which cycle you are getting a CT value is more important. And when you get a CT value after 35 to 40, we run a 40 cycles. Between 35 to 40, it means it is negative for the particular disease. Is the question answered, madam?
Yes, sir. Yes, sir. So Cybergreen or Tacman polymerase, which is best for RT-PCR?
Madam, again, these dyes are very specific depending upon what is the genetic material to be isolated and what is the disease you're screening for. For example, when you're going on with the viruses screening for in aquaculture like white spot syndrome or a EHP, only FAM and WIC regions are used. When you're going with a COVID sample, sometimes it is TAC polymerase, sometimes it is cybergreen. Depending upon, as I told you already, it depends upon the extraction process, the type of genetic material, and the type of disease diagnosed. So these are the three criteria in which we choose the dye. Whether it's a cybergreen, TAC polymerase, FAM region, WIC region, it all depends based on these three criteria: the disease diagnosed, what is the extraction procedure, and what is the nature of the source. Is this question answered, madam?
Yes, sir. Yes, sir. It feels like microbiology, biotech, bioinformatics, etc. Is any training has been given?
Biotechnology, bioinformatics.
Any other
Madam,
training program has been given? Like,
what is the exact training? There should be a specificity in training, madam, because
yes, sir. Yes, sir.
most of the companies that are in Chennai, they don't give a proper training. If you give a samples also, we exactly don't know whether the results what they are giving are correct or not. The students are not allowed to do in training in most of the smaller firms in Chennai, because they don't have a proper machinery, first one, and they do have a tie-up with other companies as a third source party. So most of the people, there are no such trainings available. Microbiology, the basics like streaking methods, how to prepare a media, and all these things is being taught in the college. Especially if you need training something like FTIR, GCMS, HPLC, the SRM University. I'm not SRM. The Satyabama University is conducting these programs yearly once. So.
Okay, sir. Sir,
Sir,
RT-PCR.
actually, ma'am, 1 minute. Sir,
Yes,
actually, the student is trying to ask in your institute, is there is any kind
Hmm.
of this training is been given? Hello, sir. Could you hear me?
Sir, you have to unmute yourself, sir. Vishnu, sir.
Ma'am,
Yes, sir. Yes, sir.
ma'am, just collect the questions from the chat box, which is available till now.
Yes, sir. Yes, sir.
We will discuss with him and we'll post the answers for these questions in a group to
Yes,
let
sir.
the candidate know, sir. We
Yes,
can
sir.
proceed with vote of thanks.
Yes, sir. Yes, sir. Now I would like to invite my colleague, Dr. Rajendran, Assistant Professor from the Department of Biotechnology, to give the vote of thanks. Sir?
Yes, madam. Madam, can you hear my voice?
I think again, I am able to hear you, ma'am.
Yes, sir. Sir?
Yes, ma'am. Hello.
Yes, sir. Yes, sir. We can able to hear from you, sir. Hello, sir.
Madam, if you can give my number, if can participate have any doubts, they or talk through my number.
Yes, sir.
Maybe
Yes,
after
sir.
7 o'clock. So I think there is some problem with the issue.
Yes, sir. Definitely, sir.
Okay,
We'll
ma'am.
Yes, sir.
Anything is very problem, not a problem.
Yes, sir. Sure, sir.
Thank you. Thank you.
Uh,
I hope the session was useful to everyone.
Yes, sir. Definitely, sir. It was a wonderful session that we had actually today. And now I request Dr. Rajendran to propose the vote of thanks. Rajendran, sir?
Yes, madam. Can you hear my voice, madam?
Yes, sir. Yes, sir. We can able to hear from you, sir.
Technology on behalf of Biotechnology, and express my vote of thanks. So, my most sincere thanks to the God for making today's event a successful one. I would like to thank our today's guest, Dr. Vishnu, Senior Scientist, Deputy General Manager, R&D, IB Group of Chennai, for accepting our invitation, sparing his time, and sharing his science and research knowledge in real-time PCR and its application. Sir has explained elaborately PCR and different types of PCR, sample preparation of PCR, two-step count of PCR reactions,
Rajendran, sir, your voice is breaking, sir. Rajendran, sir? Rajendran, sir?
Anyhow, ma'am, let it be, ma'am.
Yes, sir. Definitely,
Ma'am,
I
let us
have
hear.
no strong enough words to express my gratitude to the Dr. G. Vishnukiran Manam sir for kindly accepting our invitation and deliver the talk. We hope this professional development program would help us to expand our knowledge in the real-time PCR and the principle and the technical issue if you found like how you have to solve and all these things like sir was briefly was explained, and it was a wonderful session that we had. Thank you, sir. Thank you for
Madam, can-
joining us today to spare your time on this occasion, sir.
Madam, can you
Thank
hear
you,
my
madam. Thank
voice?
you, madam.
Hello.
It's okay, sir, ma'am. We will wind up the session and the participants. The feedback link has been given in the chat box. Kindly have a look and fill it up. And we are really apologize for the technical issues which has happened from our side. We know that some of your time has been delayed or been wasted due to these technical issues, but it's out of our control. So hope you all understand the situation, and I thank you one and all for the participation and kind patience for the entire presentation. I thank our resource person Vishnukiran Manam sir for his effort to resolve the technical issues to deliver the commitment what he was given to us. Thank you so much, sir.
Thank you, sir. Thank you, sir.
Thank you so much, sir. And the feedback link is available in the chat box. I request the participants to kindly fill the feedback link.
The resource person mail ID has been given in the chat box by the resource person. So if anybody have a doubts, you can contact to that mail ID at his convenient time. Thank you. Ma'am, we can stop recording and let the session alive for feedback. Few more minutes.
Yes, sir. Yes, sir. The feedback link is available in the chat box.